So now we are going to go into the small intestines. Remember, small intestines is also simple columnar, and the small intestines is the major site of both chemical digestion and absorption. It is 21 feet long. It has 3 segments that you need to know. The first one we've already talked about is the duodenum. This first section is only 10 inches long. And then we have the jejunum. Say that word, jejunum, jejunum. This is 8 feet long. And then we have the ileum. The ileum is 12 feet long, and it is going to be connecting to the large intestines. So 21 feet of small intestines. So I just drew some drawings so you can kind of understand the next concept better. Hopefully my drawings aren't they're pretty bad, but let's see if you can work through this with me. So we said in the small intestines is where most absorption takes place. We are going to have folds that will increase the surface area. More surface area that we have, the more area we have for absorption of our food. So this you already know. We have our 4 layers. We have our lining epithelium, lamina propria, muscularis mucosa. This is all your mucosa, right? Then we have our submucosa and our muscularis externa, the inner circular and outer longitudinal of the muscularis externa. So this is just my drawing of it. This is simple columnar. Now, in the small intestines, we are going to have simple columnar with microvilli. So you remember microvilli from week 1 of anatomy? These microvilli are cytoplasmic extensions of the cell. Simple columnar with cytoplasmic extensions. These increase the surface area. Food's going across here. We have more surface area to absorb that food. So far, so good? So that's one of the things that we have in the small intestines, microvilli. Now we are going to create a fold. We are going to have the lamina propria fold up into our lining epithelium of simple columnar with microvilli. So we're going to have these folds, and now we have our simple columnar with microvilli on these folds created by the lamina propria. So can you see how this is increasing the surface area? Again, we went from here to here. Look at all this surface area. This is all surface area, so food is going to go around each of these structures. Now these structures, we have 2 of them here that I drew. These are called intestinal villi. They are the core of an intestinal villi is that lamina propria, and they're surrounded by the simple columnar with microvilli. So far, so good? So here is that same structure I just showed you. Now we're going to create another fold. This is where it kind of gets hard to visualize. We are going to have a folding up of our submucosa now. And when that happens, now we have, here's our submucosa. It's now folded up. So now we have these intestinal villi. Here's an intestinal villi right here. This is our lining simple columnar with microvilli. This is the lamina propria. So these are all intestinal villi. So hopefully you can visualize these are intestinal villi. We just have these now. This is intestinal villi here. We just now have an infolding of that submucosa. This infolding with these intestinal villi, this whole thing is called a plicae circularis. So I drew two of them here. So in a plicae, the core is going to be the submucosa. And you can easily see this is the muscularis mucosa, right? Muscularis mucosa. So this is all mucosa. Hopefully you can see that. Here is your muscularis externa. It is not involved in the folds at all. So when you open up the jejunum, you open it up. This is what it's going to look like this. These plicae circularis, they're also called circular folds. If you want to call this circular fold, that's fine with me. These are macroscopic. These are easy to see. Easy to see. You can see them with your naked eye. They're easy to see. So this is the drawing. Here's they're showing you the jejunum. You open it up, you see these are circular folds, these plicae. Easy to see. So here we have the dark red line. That is our muscularis mucosa. So this is all going to be the mucosa. So we have, this is an intestinal villi with, this is the lamina propria, and then lining this intestinal villi is going to be the simple columnar with microvilli. So they have 1, 2, 3 plicae circularises here are circular folds. This is submucosa. Muscularis mucosa. Easy to see, submucosa. And here is our muscularis externa, not involved in the folding. Inner circular, outer longitudinal, and then our serosa. Let me get this over a little bit better here. If you can see this, here is a blow up of one of these intestinal villi. So they are blowing this up and showing you one intestinal villi. Looks almost just like my drawing. Here is the simple columnar with microvilli. And I forgot to mention, there is lots of goblet cells in here also, making their mucus. So lots of goblets. And this is the lamina propria. Now within this lamina propria is going to be capillaries. That is how we're going to be absorbing all our food. The food is up here, right? This is the lumen up here. Food's floating by. Food is floating by. We're going to absorb the food through those simple columnar and into the capillaries. We didn't talk about this. I'll just mention it. This is a lacteal. This is part of the lymphatic system that we didn't have time to get to. The lymphatic system, this lacteal is actually where fat is going to be absorbed. Fat is not directly absorbed into the capillaries. It's absorbed into the lacteals. So what does this look like? What do these plicae circularis guys look like, our circular folds? What do they look like? So I took a picture of a slide from the classroom. This is a picture, a slide of the jejunum. You can see the plicae. Look at that. You can see 1, 2, and a start of a third one. These are macroscopic. You can see them with your own eyes. The intestinal villi are coming right off of them. You can kind of see them here. This is going to be submucosa in here. Submucosa. I don't know if you can tell, but the dark part down here, this is the muscularis externa, and here's the serosa. You can see all four layers right on this, just looking at the slide. That is way cool. So when you put this under the microscope, we're looking at one of these guys, one of these plicae. This is what you are seeing. Nothing else looks like this. First thing, here is your muscularis mucosa. You can follow that muscularis mucosa all the way around. Easy to see. These are the intestinal villi. What are intestinal villi? They have a core of lamina propria, and they're surrounded by the simple columnar with microvilli. These are all intestinal villi. Submucosa, and then you can see the muscularis externa here. Hopefully this makes sense. If not, look back at this video and try to make sense of it. This most definitely will be on an exam. Now we are looking at these are the intestinal villi up close. So we are zooming in on one of these guys. We are zooming in. We are going on high power and looking at one of these guys. This is what you're seeing. Here's an intestinal villi right here. These are all intestinal villi, but this one's really clear. We have simple columnar. Here is the lamina propria. The core is the lamina propria, where we have our capillaries and the lacteals in there. Here we can see the goblet cells, all these white cells, goblet cells making mucus. This here, this is all mucus made by those goblet cells. So hopefully those intestinal villi, you can understand what they are and understand what a plicae circularis is and can identify them under the microscope. So there it is. This is intestinal villi, simple columnar, lots of goblet cells, core of lamina propria. So let's go back to our duodenum. We looked at the histology of the small intestines. There's 3 things that the intestine has that increases surface area. What are they? Microvilli on our simple columnar. Intestinal villi, which is an infolding of the lamina propria. And then we have our plicae circularis. Those are the 3 things that are found in the small intestines to increase surface area. So let's get back to our first part of our small intestines, the duodenum. So here we have the stomach, and we had the pyloric sphincter was right here. It's going to be releasing that chyme. Remember, we churned the bolus to chyme. We're going to release the chyme into the duodenum. And you can actually see the circular folds or the plicae right here too. We are going to release that chyme into the duodenum. And there's going to be 2 ducts that are coming together and secreting their content into the duodenum. One of those ducts is going to be the common bile duct coming from the gallbladder and liver. We'll talk about that more in detail when we get to the liver and gallbladder. But this common bile duct is carrying bile. And then we have the pancreatic duct coming from the pancreas. The pancreatic duct is carrying pancreatic digestive enzymes. These two ducts are going to join together, and they're going to enter the duodenum right here. Now this is on your worksheet too, so you can stop the video and do this too. Here's the common bile duct. Here's the pancreatic duct. They are joining together to form the hepatopancreatic ampulla. An ampulla has something to do with a Roman jug that has two, I don't remember, but it has something to do with a jug with two parts that come together. So this is named after that Roman jug, ampulla. Hepato because it's coming from the liver, pancreatic because it's coming from the pancreas. So the pancreas is both an endocrine and exocrine gland. This is the exocrine gland part. Remember, exocrine glands secrete their secretions through ducts. So the pancreas, pancreatic duct, bile duct join to form the hepatopancreatic ampulla, and then they're going to be dumping their secretions into the duodenum. Just one other picture, because I like this one, because it's actually showing you the dripping of the bile and the pancreatic juices into the duodenum. So I just like that. And that's why if you ever have had dry heaving and you are coughing, are vomiting so hard, you get bile backed up into the stomach and you'll actually vomit up bile. It's because the bile is coming in real close to the stomach. Just in case you wanted to know. So here it is on your classroom model. This is the pancreas. This is the duodenum, the first part of the small intestines, 10 inches long. Here's the pancreatic duct. We do not see the common bile duct, but pancreatic duct. And with the pancreas, we're going to talk about that in more detail too. This is the head of the pancreas. The head, body, and tail. So this is going to be the head of the pancreas, the body of the pancreas, and the tail is out here. So the duodenum is real important because if you do not have these digestive enzymes coming in here, it is almost impossible to digest your food into absorbable units. You don't have the pancreas, you cannot digest your foods. This is a major, major way of digesting all the foods that we eat. I think there's like 22 different enzymes. I'll have to check on that coming from the pancreas. So we've gone through the duodenum. We got our bile coming in from our common bile duct. We got our pancreatic enzymes coming in from the pancreatic duct and dumping into the duodenum. Now we're on our way to the ileum and jejunum. We've got lots of area to absorb all that food, 20-some feet to absorb all that food. Then we're going to get down to the distal end of the small intestines. So remember we said duodenum, jejunum, and then the ileum. So the distal ileum is going to be connecting to the first part of the large intestines. This area where it's dumping, this is still called chyme, into the large intestines. This is called the cecum. Cecum. So here is our distal ileum, the last part of our small intestines, dumping the chyme, the food. Well, hopefully most of the nutrients have been absorbed, but dumping that chyme into this part of the large intestines called the cecum. This is a pretty It's a round little ball down here, the cecum. The cecum, there's a valve that controls the flow of the chyme into the cecum. It's called the ileocecal valve. Ileo coming from the ileum, cecal going to the cecum, ileocecal valve. We want our chyme to be going in this direction only, into the cecum. We do not want the contents of the large intestines backwashing or going backwards into our ileum. Not good. The cecum is also where you have your appendix. So that is just lymphoid tissue. Lots of white blood cells are in the appendix. So here's the appendix here. This is going to be in your right lower quadrant. So the right lower quadrant pain, you always think about the appendix. So I wanted to show you something that happens at the distal ileum that is, to me, way cool. So most of our plicae, the really big plicae circularis, that's going to be in the jejunum. So that slide I showed you, that was the jejunum. So when we get towards the distal end of the ileum, remember the ileum is 12 feet long. So towards the distal end of the ileum, the part that's getting closer to the cecum, we're going to have a change. Number one, we're not going to be seeing plicae. We still see lots of an intestinal villi. These are all still intestinal villi. Lots of intestinal villi, but just do not see the plicae. Here is our muscularis mucosa. Easy to see. So this is all mucosa. Here's our submucosa right here. There is nothing in this submucosa. Nothing, 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 nothing. Then bam, we are now reaching the distal end of the ileum. We are going to be running into these guys, Peyer's patches. This is lymphoid tissue, lymphoid nodules. So what are Peyer's patches that you need to know? They form an important part of the immune system. They monitor intestinal bacteria populations. Lots of potential for bacteria in here. And prevent the growth of pathogenic bacteria in the intestines. Now there's good bacteria in your gut, and there's bad bacteria. These are monitoring the bad bacteria that can kill you. So, important part of your immune system. This slide is from our classroom. I think it's one of the most beautiful slides I've ever seen. You won't find a slide this good on the internet. Easy to see the mucosa. Easy to see that muscularis mucosa, submucosa. Look at that muscularis externa. You can see the two layers, two different colors, inner circular, outer longitudinal. And those Peyer's patches are just beautiful. So Peyer's patches, know what they are and where you find them and why they're there. This is from the internet, so you can see their Peyer's patches. They're good too. They're good. They're good. Not as good as ours, but they're good. Here's your muscularis mucosa. This is all intestinal villi here. This is their submucosa. These Peyer's patches are in the submucosa. And here's the muscularis externa. Same thing here. Here is the Peyer's patches in that submucosa. So if you see these, and I say, where are you in the GI tract, and you see a Peyer's patch, what are you going to say? You're in the distal ileum. So we are now into the large intestines. We're almost done. And it's almost an hour video. Oh my God. So we are in the cecum. We went from the chyme from the ileum, went into the cecum through that ileocecal valve. Now, what do we need to know about the large intestines? It's 5 feet long. Its diameter is 2.5 inches. I don't know if I mentioned it, but the small intestines, the diameter of the small intestines is 1 inch. So we have 4 parts of the large intestines. The first part is this, the cecum. So the cecum is the first part that has the appendix. Then we have the colon. Now we're going to have different parts of the colon. We have the ascending colon, goes up, transverse colon goes across, descending colon goes down, sigmoid colon makes this S shape, and then we have the rectum. Remember, rectum is straight, so we got this straight part of the large intestines. And then we have the anal canal. The anal canal, the last 1 inch of the rectum, that is going to be stratified squamous again. Everything else here is simple columnar. So there are some special features on the large intestines that you need to know. One of them is called the Tinea coli. Now you're going to see this spelled differently too, but I spell it this way. You can spell it this way. It's a legal way to spell it. Tinea coli. What are Tinea coli? If you see these little bands here that are running across on both sides of the large intestines, this is the Tinea coli. What are they? These are the outer longitudinal muscle fibers of the muscularis externa. Instead of having the muscularis externa wrap all the way around the large intestines, you are just going to have these bands of fibers that are going to form the Tinea coli. But this is the part of the longitudinal muscle. You still have the inner circular muscle going around, but the longitudinal muscle is now just this band called the Tinea coli. And it extends the length on both sides of the large intestines. You're going to lose it once you get down to the rectum. And then you're going to have these pouches. These pouches are called haustra. So you need to know those pouches too. So they are formed by the tension of these longitudinal muscles that are pulling on this tube. It's just like if you were sewing a piece of material and you put your thread in and out of the material and then you pulled that thread, what would happen to the material, that material the thread was in? It would bunch up. That's exactly what's happening up here. You're getting this bunching up of the large intestines, and you're making these little pouches. These are called haustra. I think that's it. Here's the appendix again. Here's the showing you the ileocecal valve and the distal ileum. So we're going to look at the histology of the colon. We're almost done, guys. So first of all, you need to know there are no intestinal villi in the large intestines. There are no plicae circularis in the large intestines. Those two structures are only in the small intestines to increase surface area for absorption of all our nutrients. So what do we have in our large intestines? So here's the surface of our stomach. We know we have simple columnar because simple columnar is everywhere. We have our simple columnar cell, and then there's going to be what they call the intestinal glands. There's going to be these dips that are going down in through the lamina propria. Here's the lamina propria. Here's the muscularis mucosa. And what's in these glands? What are they secreting? These are all goblet cells. Goblet cells secreting mucus. Lots of mucus is being excreted onto the surface of the large intestines. So if you look at this, a lot of people go, oh, these look like intestinal villi. No, this is the lining simple columnar, and then you have these glands. Look how white they are. All those white dots, those are all goblet cells secreting their mucus. Why do we want so much mucus? To help lube. Remember, we were going to lube the tube to help the passage of the feces that we are now creating out. So here's your muscularis mucosa. Here's the submucosa. You see something like this, you look at all those goblet cells, you know you are in the large intestines. So we have 5 feet of large intestines. What is the function of the large intestines? Well, we're going to be absorbing most of the remaining fluid, water that's in the chyme. We're going to absorb some electrolytes and vitamin K and some B vitamins. And we're going to turn that chyme into feces. So that is the job of the large intestines. So here it is on our classroom model. Here is the cecum, ascending colon, transverse colon. You can barely see the descending colon here. This is the pink. They're showing you the tinea coli in pink here to highlight it. Here's the tinea coli here. Tinea coli all in through here. Remember, these pouches are called haustra. Haustra. Here is the distal ileum. Here's the ileocecal valve. Here is the cecum. And here's the appendix sticking out back this way. Here is the descending colon coming down here. Here's our sigmoid colon. And now the rectum is going to go down back in here. So where are we? Yay! We're coming to the end of our magical journey. This was the video of food to feces. So we are now, this is all feces. What's coming down is now all feces. So here's our sigmoid colon. This is simple columnar. Here's our rectum. Rectum, remember, rectus means straight. So this is our rectum. This is the last 8 inches of our large intestines. And then we're coming down to this junction right here. This is where we change from simple columnar to stratified squamous. This last 1 inch is the anal canal. The other thing you need to know is we have smooth muscle sphincters. Here's the smooth muscle. This inner smooth muscle is the internal sphincter on each side. The internal sphincters, smooth muscles, are controlled by the autonomic nervous system. Parasympathetic will relax them so you can poop. Sympathetic will contract them so you don't poop. This outer one is the external anal sphincter on both sides. This is skeletal muscle. This is the one that you control to hold the feces in. And most of the time it does work. So anything else? I think. Yeah, there's when you defecate, that's where it all ends. You have a reflex when this rectum is a holding tank. When it stretches, you are getting a reflex signal sent to your brain those stretch receptors are going to signal your brain that you need to go poop. So we're done. So remember, you are what you eat. Here's our little froggy. He's filled with light. So we will have one more digestive system video that will just look at the gallbladder, liver, and pancreas.